A-Level Biology 9700, Paper 4, Variant 1, May June 2025. Question 10 or last question of this paper. And question 10 is, during the course of an action potential, the potential difference across a neuron membrane changes. Table 10.1 shows the potential difference across the neuron membrane at three sequential time points X, Y, and Z just before and during an action potential. So you can see in table 10.1, the time points are X, Y, and Z, and at X, the potential difference is minus 70, and at Y it is plus 40, and at Z it is minus 90. So describe the events that cause each of the potential difference at these points X, Y, and Z. So X, Y, and Z, uh, the different events which cause during this process, that is the when there is X means it is minus 70. So the potential difference which will be at minus 70, the neuron is at its resting membrane potential. So the membrane is polarized at this time. The state is maintained by the sodium potassium pumps actively transporting 3 sodium ions out for every 2 potassium ions in, and the membrane being more permeable to potassium ions than to the sodium ions. So as a result, there is a higher concentration of sodium ions outside and potassium ions inside. So the axon leading to a negative potential inside relative to the outside. So then the at time point Y, which is plus 40, you can see the peak of action potential is here at this point. So due to which the membrane is fully depolarized, reaching the peak of the action potential. So a stimulus, why it is reached at this position, a stimulus has caused the membrane potential to reach the threshold potential, leading to the rapid and massive opening of voltage gated channels for sodium ions. So as a result, the opening of these channels, the sodium ions rapidly flood into the axon down their electrochemical gradient, causing the potential inside the axon to become positive. So the voltage gated channels for sodium then quickly inactivate or close. Then the time point Z is at minus 70. It means the hyperpolarization. So at minus 90, the membrane is hyperpolarized. At 70, it is hyperpolarized. And during hyperpolarized, what happens? The inside of the uh, axon becomes excessively negative, and as a result, the potassium channels finally close, and the sodium potassium pump along with potassium plus leak quickly restores the membrane to the resting potential minus 70. So I would like to write, as I have written for minus 90, minus 70, the membrane is polarized. And at minus 40, the membrane is depolarized. So these are the three sequential differences in the potential difference. First of all, at X, it is polarized. At Y, it is depolarized. And at Z, it is hyperpolarized. So because it is minus more than the minus 70. So at positive, it is depolarized. That is plus 40. So I would like to write all this explanation which I have told you about this. So the neuron is at its resting uh, at X. This state is resting potential which is maintained by sodium potassium pump actively transporting three sodium ions three sodium three sodium ions out for every two potassium ions in in. As a result, there is high concentration, high concentration of sodium outside and potassium ions inside, leading to negative potential inside relative to the outside. Outside. And the second point is plus 40 means this is the peak of the action potential. The membrane is fully depolarized. This is the first, and at time point y, the membrane is fully depolarized. Is depolarized reaching the peak of action potential. So which leads to rapid and massive opening, rapid and massive opening of voltage gated channels. As a result, sodium ions rapidly flood into the axon down their electrochemical gradient, causing the potential inside the axon inside the axon to become positive. The voltage gated channels then quickly close. Now the time point, third point, which is the time point C. So what happens over there? The membrane highly depolarized. Sorry, highly hyperpolarized. Means the potential is more negative than the resting. Is more negative than the resting potential, that is minus 70. The voltage gated gates will have opened, that due to which it happens. So the cause was opening of voltage gated channels, potassium channels Then so in response, to depolarization, now allowing allowing K positive ions rapidly to leave exon. Down their electrochemical gradient. So the inside of the exon becomes excessively negative. That is minus 90. And then K positive channels finally closed. So it was part A. Now we will move the second part of this question. And second part of this question is suggest how one action potential causes an action potential in an adjacent section of the axon of an unmyelinated neuron. So here the process, we can say that this process when there is an action potential cause an action potential in the adjacent section of the axon of the unmyelinated neuron. So this process is called local current flow or conduction by continuous propagation. So this process is called as local current flow or conduction by continuous propagation. So how do this take place? So first of all, there will be the depolarization. So an action potential is currently occurring in the first section of the axon. The inside of this section is momentarily positive due to the inrush of positive ions through open voltage gated sodium ions. So then the high concentration of positive charge is the cytoplasm of this section repels positive ions in the adjacent resulting resting section of the exon. This creates a local electrical flow that flows from the depolarized region to the adjacent resting potential. So this movement of change causes the membrane potential in the other section to become slightly less negative. So if the local current flow is strong enough, it will cause the membrane potential to reach the threshold potential. So reaching the threshold potential causing the voltage gated channels in the other part of the section to snap open. So the subsequent rapid influx of the ions into initiates a new action potential in the new that adjacent section. So this process is then repeated. The newly depolarized section B, which generates a local current that causes the next sections to siege in stretch, so on. In this way, the action potential is propagated continuously entire length of the unmyelinated Keegan. And directionally, crucially, the action potential can only propagate in one direction. So the section of axon that just completed the is its refractory period, and then the absolutely refractory period, which where its voltage gated channels are inactivated and cannot be opened regardless of the stimulus. So this prevents the impulse from flowing backward. Would you like uh, to? We can also compare all these the different sequence which are taking place. So how now we have to give two suggestions only. That is two suggestions we can say the takes place first of all by the uh, local current flows. The high concentration, I think we write down the sequence. An action. It's the local electrical current flow from section, first section, from section to the to, to adjacent section. So the local current flow and then the movement of charge causes the membrane potential to become slightly less negative. So threshold and initiating the new action potential. If the local current flow is strong, it will cause the membrane potential in section B in adjacent section to reach the threshold potential. adjacent section to threshold potential. SHOLD is threshold potential. So the subsequent influx of the ions into the adjacent direction in new action potential. So I would like to write over here the subsequent rapid influx of ions into adjacent section initiates a new action potential. So in this way, it can happen. Now we will move towards the last section of this question, that is C. Vertebrate animals generally have a myelin sheath. Myelin sheath, you know, the insulating sheet around the axons of motor neurons, explained by the presence of a myelin sheath around a motor neuron axon is an advantage. So we have to write the advantages of this myelin sheath around the motor neuron. So first of all, I would like to say that the myelin sheath act as an insulator. Acts as an insulator because it is composed of fatty material it is um, it is not continuous, has gaps called as nodes of Ranvier. Ranvier the jumps from one node to the next cause a process called saltatory conduction. This is much faster than the continuous conduction seen in unmyelinated axons. So it is responsible for survival and fastest fastest possible transmission of motor commands, enabling effective effective muscle action. So it was all about the question number 10. And now this paper has been completed in 8 videos. Please like, share, and subscribe my channel to watch more papers. Thanks for watching.